Hello, good morning, everyone. Let's get started. This week, we are going to be doing Lecture 6, Cellular Reproduction. This is the first lesson of the second half of the course. So everything that we did for the first half of the course, you will not be tested on them anymore. Right? Everything that is on Lecture 1 to 5 that was assessed on the test last week. And moving forward, you will only be getting tested on lecture 6 all the way to the last lecture, which is lecture 10. Okay, so there's no final exam. There's only test number 2, which again will only cover the second half of the course. So hopefully that gives you a little bit of like a sign of relief, maybe, knowing that you don't have to memorize so many things in the course. So the first lesson in the second unit, second half of the course, is cellular reproduction. All right. So if you recall from the cell theory, okay, one part of the cell theory states that all cells came from pre-existing cells, right? The cells came from the division of a pre-existing cell. So today we'll be focusing on that division. All right. So here we have what we call a parent cell. Parent cell. Okay. Now let's just, for the sake of simplicity, let's say it only has 2 chromosomes. Okay. So there is two sets of chromosomes. Okay. So there's a chromosome 1 here, and then there's chromosome 2 here. Okay. So total of 4 chromosomes. Okay. And what's going to happen is you will want to do DNA replication. Okay. And in DNA replication, the cell is going to copy all of these chromosomes. Okay, I'm just going to draw a little bit bigger so it's more clear. So now this chromosome, you're going to have an exact copy. This chromosome, we're going to have an exact copy. And then same thing for the smaller one. Okay. And here is the confusion for most people. I was confused as well. Sometimes I still get confused, to be honest. But I would like to ask, how many chromosomes do we have here? Okay, you started with 4 chromosomes. And then after DNA replication, how many chromosomes do we have now? Tyler said 4. Anyone else? Okay, Bianca also said 4. And that is correct. Right? Even though it looks like there's 8 things here. However, if they are still stuck together like that, the definition still counts that as 1 chromosome. Okay, so here's 1 chromosome, 2 chromosomes, 3 chromosomes, 4 chromosomes. Okay, so it is still 4 chromosomes. However, after DNA replication, each of these we call them a chromatid. Okay, chromatid. So even with 4 chromosomes here, we would have 8 chromatids. All right. Now, just to keep it simple, right? The chromosome counting is not too essential in understanding what's happening. What's more important is the fact that after DNA replication, you have double the amount of genetic material, right? So when you do cell division, when you do mitosis here, right? The chromosomes will separate. And once again, you will be back to 4 chromatids, right? We don't actually call them chromatids anymore since they're not really attached, but you will be back to the same amount of genetic material that you started with. Okay, so this one is the parent cell. These are 2 genetically identical daughter cells. Okay, so these are exactly the same in every way genetically as the parent cell, assuming there is no errors during the whole process. All right, so that's what we're going to be looking at today. For the first half, we'll be focusing on the DNA replication, and then for the second half, we'll be focusing on the mitosis. Okay, so 4 chromosomes again. Any question about this concept? Okay, we will go ahead and look at these scientists. So first one is Chargaff. Okay, so Chargaff has the Chargaff's rule, which basically, well, the guy, he discovered that in any DNA sequence sample, okay, the number of adenine, which is A, always equal to the number of thymine. And so this shouldn't be surprising because A pairs with T's, right? So if you have, 60,000 adenine in the DNA, you're going to expect to have 60,000 thymine as well. Okay, and of course, the second part of this statement is the number of guanine equals to number of cytosine. Okay, so based on this, we can also write out the DNA base pairing rule. Which is A pairs with T and C pairs with G. Okay, so DNA are, the 2 strands of DNA are complementary. 2 strands of DNA are complementary. Okay, that's quite simple. We kind of know this already. I hope you knew this. Okay. Next one, we have Rosalind Franklin. Sorry, Rosalind Franklin. So what did she do? Well, what she did is she used X-ray to study the structure of DNA. Right. Her data suggests DNA is a double helix. Okay, so she was not able to finish her work because she died from ovarian cancer, probably due to exposure to X-ray. They didn't have as much safety protection before as we do now. So she was not able to complete her work. And so comes Watson and Crick. Okay, they using Franklin's data, they built the first scaled DNA double helix model. Okay, like if you search up Watson and Crick in Google image, you will see the model that they built. Okay, it's a classic picture that is found in almost every textbook. All right, Hershey and Chase is next, right? So what did these 2 scientists do? Well, what they did is they used they used bacteriophage. And if you're not sure what that is a virus that infects bacteria. Okay, they look like this. Okay, they look like the little sentinels from Matrix. So they used bacteriophage to show that DNA and not protein is the hereditary material in a cell. So the DNA is the material that gets passed on. That's the blueprint of life. Okay, that encodes for all the instruction for the organism. That's the stuff that gets passed on rather than the protein. Okay, because there was this big debate, right, in the scientific community back then, whether it was DNA or protein that's responsible for encoding instruction. Protein has 20 amino acids, so it makes sense that it is the hereditary material because it had so much more coding capacity, right? Whereas DNA only has 4 bases. But then Hershey and Chase experiment definitively show that it is the DNA that's responsible for encoding the genetic material. Okay, next one, Matheson and Saul. Anybody remember what they did? I've been talking for 10 minutes now, so for a change. Does anybody want to tell me what did Matheson and Stahl do? What did they show? Okay, so they used E. coli to show that DNA replication is semi-conservative. That's too thick. Give me a second. Semi-conservative. Okay, it means when you replicate DNA, exactly one strand will be the original, and then the complementary strand will be completely new. Okay, so we can talk more about that later. I guess I can quickly show you. Right? So here is your original double helix. Okay, so after DNA replication, you're gonna have two of these copies, right? So this is the O strand, and then you will have the new strand. Okay, so this is new DNA molecule, new DNA strand. This is the original DNA strand. How's that? Okay, number 2 should be quite straightforward. You should be able to do this by now, right? The complementary strand for DNA below, be sure to include directionality. So this is ATC, GGATTA. And so the complementary strand is going to be TAG, CCT, and the top strand here goes from 3' to 5' direction. Okay, the 3' and the 5' is just talking about which carbon contains a hydroxyl group, for example. We're not going to worry about it. Okay, just know that one side is going from 3' to 5', and then the other strand goes from 5' to 3'. It is in the opposite direction. So not only are the DNA strand complementary, they are also anti-parallel. Okay, anti-parallel. That means they runs in opposite direction. Okay, and obviously they're parallel to each other. So if it's difficult to visualize, you can think of it like this. Okay, think of a bunch of people lying down on the floor. Okay, head to toe. Head to toe. Like that. Okay, so we call this head to toe. Okay, so this is like one strand of the DNA. And then on the other side, to demonstrate the concept of anti-parallel, it would be the same people, but backwards. Okay, so it's parallel in the sense that they are right next to each other. The distance between them don't change. Okay, but now instead of head to toe, we're going to have toe to head. Okay, so obviously the DNA strands don't have head or tail or toes. So instead of head to toe, we would say 3 to 5. And then on the other strand would be 5 to 3. Okay. Is that okay? Any questions? Okay. If 23% of a cell's nucleotides are adenine, we did this before as a thinking question, application question in the earlier chapters, but let's go through it again. Okay, so 23% are adenine, what percentage of thymine, cytosine, and guanine? Okay, so if you have 23% adenine, by Chagap's rule, you know there is going to be 23% of thymine as well. Okay. So if you add these two up, right, 23 plus 23, that's going to give you a total of 46%. Okay. So 46% will be adenine and thymine all together. Now, if I take 100% minus the 46, I will get 54%. Okay, so this 54 accounts for the C's and the G's, right? Okay, so if I divide it by 2, I divide 54 by 2, I would get 27% C and 27% G. Okay, so that's how you get the answers. All right. List the 4 stages of cell cycle. And so we have G1, we have S, G2, and M. Okay, so this here, the first 3 letters, these are the interface. Right? The cell spends most of its time in interface, performing its function, growing, gathering nutrients. And finally, the M here, that is the mitotic phase or mitosis, right? The actual cell division. Okay. So what is the significance of G1 and G2? G1 and G2 are checkpoints, okay? The cell is going to go through a series of checklists, basically, to make sure that the cell is in good shape to proceed to mitosis. Okay, so during the S phase, that is where the actual DNA replication occurs. But before you copy the DNA, before you invest the energy into copying the DNA, you're gonna want to do a little bit of check. So, for example, in G1, we can check for things like cell size. Okay, cell size. Is the cell large enough to divide? Okay. One of the reasons why we have to do cell division is as the cell gets bigger and bigger and bigger, the surface area to volume ratio becomes not optimized. Okay, there's going to be a point where there's too much volume and not enough cell membrane to allow the cell to do its job efficiently. So you must divide. So one of the things you want to check for in G1 is the cell big enough to divide? Okay. You don't want a small cell to divide, or you're not going to have enough cytoplasm to split. Okay, so that's one thing to check for. Okay, you're going to check for nutrients, right? Is there enough resources, enough energy plus resources to divide? Okay, you're gonna be doubling all your genetic material. Do you have enough nucleotides, right? Do you have enough ATP, right? To kind of fuel this process, right? If you don't have enough of either, then it's not a good idea to divide at this point. Okay, so there are other things like a molecular signal. Molecular signals. Okay, so example of a molecular signal is like growth factor, right? Growth factor. There are growth factors that would encourage cell division. If you think about like a baby growing, right, they're gonna need a lot more cells to get bigger and bigger, right? Like as a person, right? So there are gonna be a lot of growth factors that stimulate that. Okay, the opposite of growth factors would be, growth suppressor. Okay, which would put a brake on the cell cycle. So there's a balance of these molecular signals, and G1 checks for those. Right? Obviously, we do not want too much stimulation from growth factor, especially in adults, for example, because if the cell cycle keeps on going in an uncontrolled manner, that could lead to creation of a tumor, right? And the tumor could be cancerous. And finally, one last thing, we want to check for DNA integrity. DNA integrity. Okay, are there any DNA damages? Okay, can they be repaired? Right. If not, then it's not a good idea to divide the cells. You don't want to be copying a cell when there's a lot of errors in the DNA, right? So all these checking is primarily done by proteins. Okay, so if the checkpoint proteins are mutated, then that means you might not be able to accurately assess these situations. Okay, and that could lead to a situation where, despite the fact that you don't have enough energy or resources, despite the fact that there's no growth factors present, you still allow the cell to divide. Okay, and that is, of course, like a bad thing to happen, right? Okay, so just to reiterate, all these things are done by checkpoint proteins. So if the checkpoint proteins are mutated, then you might not be able to detect these things, and you let the cell divide when it shouldn't have. Okay. So if all goes well, no problem, then we are going to allow the cell to copy all of its DNA. And then after DNA has been copied, we're going to go for a second checkpoint, okay, G2. It's like getting your driver's license, right? You go through G1, and then now you got to go through G2. So G2, we check for DNA integrity. Okay. Again, are there any damage? Are there any DNA damage? Because when you copy the DNA, you got to unwind them, you got to wind them back up, right? All that moving around the DNA could have caused some damage, right? Maybe you make some mistakes, right? So we got to be careful here. We got to check it. And then we also want to check for DNA errors. Okay. Can errors be corrected? Okay. And if not, then again, the cell is not allowed to move on to mitosis. Okay, so that's your G1 and G2. So let me just put like one last point here at the bottom. If the cell fails G1 or G2, it might trigger apoptosis, right? Apoptosis is programmed cell death. Right? Remember those lysosomes? They could release all of its content and make the cells go kaboom and terminate the cells. Or if apoptosis is not the suitable choice, we can trigger growth arrest. Okay. Another word for growth arrest is senescence. Okay, and that's one of the theory of why we age. Okay? When we get older, eventually the cell's going to stop dividing, and if you have a large pool of cells that are in growth arrest, that are in the senescence state, that contributes to the process of aging. All right, I've talked a lot again, and any questions at all? Everybody okay? All right, I hope so. Let's do we have any polls here? Not yet. Okay, these are for later. Okay, let's keep going then. Okay, list 4 stages of mitosis. That's pretty straightforward, I hope. We got a prophase. And after prophase, we have metaphase. That's right, Adriana. Anaphase. And finally, telophase. Okay, so PMET. You can remember that. Okay, next, let's complete the chart below, describing the function of various enzymes and proteins and process of DNA replication. So these are all enzymes that are necessary for the process of copying all your DNA. Okay, so we start with gyrase. The purpose of gyrase, what is it? Okay, so gyrase is an enzyme that relieve tension in the DNA strand. As it is being unwind. Okay. And this is important because it prevents the DNA from breaking. If there's too much tension in the DNA as you unwind it and you don't relieve the tension, it's just gonna break. Okay, so to relieve the tension, they actually create little cuts in the sugar phosphate backbone here and there, and that will help alleviate the tension. Okay, next one. What is the next one? Next one is helicase. Okay, so helicase is the enzyme that would do the actual unwinding. Okay, unwinds the DNA to create a replication bubble. Replication bubble. Okay. It travels into the replication fork. As DNA is being copied. Okay. So here it is again. We have our double helix. I'm not gonna draw the twisted up version. It's like too difficult to follow, probably. So here, this is the 2 strands. We have the 5 prime, 3 prime, 3 prime, 5 prime. And then what's gonna happen is we're gonna have our helicase. Helicase is gonna come along and then it's gonna open up the DNA. Okay, let's put another helicase here. You don't really have to draw this. You can just watch. And it's going to create a replication bubble. Okay. So this here, this is a replication bubble. Okay, and then the helicase will keep on unzipping. Think of like the little zipper you have, right, in the jacket, right? This is basically, I don't know what you call that thing. That's the thing that you pull up and down to zip and unzip, okay? So this is gonna go into the replication fork, okay? This area here is the fork, okay? There is another fork here, okay? And then you will have all your base pair in between, right? Okay, so like, you know, T, T, A, C, and then on the other side would be A, T, G, for example. Okay, so that is the job of helicase, right? And as you unwind the DNA, right, this is the rest of the DNA strands, right? Then tension is going to build up later on here, and that's why we need the gyrates to come along and relieve the tension. Otherwise, this is going to get like all crumble up in a knot, and it's going to break off. And we do not want that. Now, there's another thing. When you unzip the DNA, they actually want to come back together because the T is attracted to the A, right, with the hydrogen bonds. And then the C and the G's are also attracted to each other through the hydrogen bonds. And they have a tendency to come back together. They have a tendency to re-anneal. Okay, that's the proper term. So we don't want that, right? Which is why we have the single-stranded binding proteins. Okay, so the single-stranded binding proteins will stick to the side here and prevent them from coming back together. Single-strand binding protein. Okay, so the purpose of a single-strand binding protein is to binds to the single DNA strand to prevent re-annealing. Okay, prevent them from coming back together. Okay. The backbone, you can say, the backbone of single-stranded DNA, single-stranded DNA, sorry. Okay, and so they're not going to come back together. Hashana said, can you explain the helicase function again? Okay, so helicase, right? Something has to unzip the DNA, right? The DNA are usually together, right? The two strands. If you think about it, right? That's one strand of the DNA. That's the other strand, and then the nucleotides, the A's and the T's, are what's holding the two strands together, right? So it's very difficult to copy the DNA when they're together, right? So something has to unzip the DNA, something has to open the DNA, and that is the job of helicase, okay? And by opening it up, right, you create a replication bubble, then you can gain access to the nucleotides. That's basically, in its simplest sense, what it does. Perfect. Okay, so what's next? Well, then we have these things called primates. Okay, now I'm gonna skip primates for now, and then I'm just gonna do DNA polymerase 3 first, and then I'm gonna come back to primates. Okay, and you will know why in a second. Let me zoom in here. So DNA polymerase is an enzyme that is going to use the parent DNA, as a template to build a complementary strand. Okay. So once you open it up, if you see A, T, C, then DNA polymerase 3 will know, okay, A pairs with T, so I'm going to add a T. Okay. And then, you know, T pairs with A, so on and so forth. It's going to build a complementary strand. Okay, so just in case you don't quite understand, I'm going to draw this again, right? We're going to have A, T, C, C, G here. And then on the other strand, you don't have to copy this. You can just watch, okay? So 5 prime, 3 prime, 3 prime, 5 prime. Okay, so what happens is the DNA polymerase 3 is going to look at each nucleotide. Okay, it's like there's a C here, so the new strand should be a G. New strand should be a G and C. Same thing over here. If it sees a C, it will put a G and a C and a C. Right, so it's going to use the parent strand, right, the black one here, to build a complementary DNA strand. Okay, the pink one here, that's going to be the new strand. Okay, and it's going to be following the base pairing rule. Okay. Now there is a bit of a problem here. Okay, DNA polymerase 3 has 2 limitations. Okay, 2 limitations. Sorry, the program just froze for a second. Let me restart and see if it's going to come back. Can you guys see it? It's functioning, right? Okay. So as I was saying, there are 2 limitations for DNA polymerase 3. 2 limitations. Okay. Number 1, cannot initiate the replication process. Okay. It doesn't know where to start, right? But once you start for the DNA polymerase 3, then it will continue, but it doesn't know how to start. Okay, it's like a child coming back from school. They don't know where to start in the homework. You're gonna open the book for them, tell them, okay, start here, question number 1, and then hopefully they will automatically start from there. Okay, so something has to initiate the process for DNA polymerase 3. And then second limitation is that can only build new strands in the 5 prime to 3 prime direction. Okay, so those are the 2 limitations we have for DNA polymerase 3. Okay, so how do we solve this problem? Well, to solve the first problem, this first limitation, that's why we need primase. Okay, so primase, what it does is it will create an RNA primer that will help DNA polymerase 3 to start the replication process. Okay. Now, this was explained in the video, but I'm just going to quickly show you again what I mean. And again, you don't have to copy this unless you really want to. Okay, so this is the replication bubble, right? We have the helicase here, which I'm not going to draw because it's already very crowded. And I'm just going to make up a bunch of nucleotides. Let's say this is 5 prime to 3 prime, and this is 3 prime to 5 prime. Okay, so I'm going to have like, you know, T, T, A, C, G, totally making this up. It doesn't really matter. T, 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 A. And then on the other strand, it's going to be complementary. Okay, so let me know if I make a mistake. I am trying to not make a mistake, but all these A's and T's can be challenging. All right, so what happens is we have this enzyme called primase. Okay, primase. Let's make it into like a purple thing here. Primase. Okay, so primase will come along. Okay, and then it's going to actually, I'm going to put it right here. There's going to be a primase here. Okay, and then there might be another primase here. Okay, and the function of the primase is to add an RNA primer. Okay, so here it's going to read the parent strand nucleotide AAT. So it's going to put an RNA primer. And there's no T's on RNA. That's why it's going to be a UUA, right? So there you have a short RNA primer. Okay, and again, this other guy right here, this other thing, that is the RNA or like just primase. Okay, so the function of the primase is to put a bit of an RNA primer. And we're going to do the same thing on the other strand. Okay, so this is GTA. So the RNA primer is going to read CAU. Okay, so that is going to help our DNA polymerase. So when the actual DNA polymerase comes, I'm just going to draw this big triangle thing. This is the DNA polymerase 3, right? It will now know where to start. It's like, oh, okay, that's the RNA primer. And I'm just going to continue building from that point on. Okay, so UUA, and then it's just keep going. AAT now, because we are building DNA, GGC, so on and so forth. It's just going to keep on going and going in this direction. Okay, now pay attention here. You can see the original strand is from 5' to 3'. And because DNA is anti-parallel, this new strand that we're building is actually going to be 5 to 3, right? It's opposite to the parent, right? 3 to 5 in the parent, and then here is 5 to 3. And that's okay for DNA polymerase, because if you look at what we wrote down earlier, it can only create strands from 5 to 3 anyways. So it's going to, as the replication bubble gets bigger and bigger, as helicase opens up the fork more, the top strand here will be going to keep on growing and growing. That is not the same case for the bottom. Okay, so here we're going to have a DNA polymerase 3 again. Now, which way is the DNA polymerase 3 going to go in this case? Is it going to go to the right or is it going to go to the left? Okay. Let's just do a poll and see if you guys are all with me. My question is, will this DNA polymerase 3 go to the right or go to the left? Okay. If you think it's to the right, please choose A. To the left, please choose B. I guess the way I've drawn it kind of gives it away, but just want to see if you guys are with me, you know. Okay, well, most of you got it right. It is going to be going to the right because if the bottom strand is 3 to 5, then the new strand is going to be 5 over here and 3 in this direction, right? So the DNA polymerase 3 can only build from 5 to 3 on the new strand. So it's going to go, you know, CUA is already there, and it's just going to add more TT, A, so on and so forth, and it's going to grow into the fork here. All right. Now that is a little bit of a problem because how are we going to replicate this stuff? Okay, how are we going to replicate this stuff? Right, because I cannot go backwards here, right? So what do? Well, what we can do is we are just going to need to have an other RNA primer. Okay, so there's going to be the primase. There's going to be another primase maybe. Okay, and then they're going to put down more primers. Right, so there's going to be like a GUA here. Okay, and then there's going to be maybe another primer here, right? I'm just going to make up a few more. GC or something. Okay, and then you're going to build more and more. Okay, so this top strand that is being produced continuously, we call that the leading strand. And then over here, the strand that is being synthesized in fragments, we call that the lagging strand. Okay, and these fragments of DNA, those are Okazaki. Okazaki fragments. Does anybody have any questions about any of these things that I've explained it so far? Are you sure? Okay, thank you. So let's go back to here, right? Okay, now we explain what the primase does. The primase put the primer in place so that the DNA polymerase 3 will know where to start. All right, and then once that happens, then the polymerase 3 will take over, and it will either produce it directly into the fork on the leading strand or in fragments on the lagging strand. Okay, so let's take a look at this picture again. Right, eventually there's going to be a DNA polymerase 3 here as well, right? I did not draw it, but it's going to be here, and it's going to keep on growing. So A, G, G, C, and eventually it's going to reach the RNA primer that was laid before, right? And at that point, when you reach the RNA primer, the DNA polymerase 3 will now fall off, okay? And then another polymerase will come to finish the job, okay? So that is the DNA polymerase 1. Okay, the function of DNA polymerase 1 is to chop off these primer because we cannot have a uracil in the DNA, so they're gonna replace the primer with actual DNA nucleotide. Okay, so the C is fine, it just stay as a C, the A will stay as an A, and then it will replace the U with a T. Okay, so that is the job of the DNA polymerase 1. Let me write it down for you. And again, all this thing is in your notes, right? We are just summarizing it, right? We're just having a very in-depth review, I guess. So DNA polymerase 1 will replace DNA polymerase 3 when DNA polymerase 3 approaches an RNA primer. Okay. And then the DNA polymerase 1 will replace the RNA primer with DNA nucleotide. Okay. Now I should also mention at this point that both DNA polymerase 3 and 1, they both have spell checking mechanism. All right. So if it finds out that it puts in the wrong nucleotide, it's able to kind of go back a little bit and take out the wrong one and then replace it with a new one. It's how sometimes we type, right? We type very fast, and then sometimes we realize we made a mistake. We just press backspace and then make the changes, right? It's kind of like that. Okay. So just two more enzymes. We have the ligase. The function of the ligase is to join the Okazaki fragments together. Okay. So because the lagging strand is produced in fragments, we need to connect them together. We need to make sure that we create that sugar phosphate bond, right, between the nucleotides, and that way we ensure we have a complete continuous strand of DNA. Okay. And then there is something called telomerase. Okay. And I should explain what telomerase is. This is only found in germ cells. Germ cells are the cells that becomes sperm and egg, okay? And stem cells. Okay. So what do we need the telomerase for, and why is it important? Allow me to explain in this diagram again. Okay. So I'm just gonna draw like a new one, not entirely new, but kind of related. Okay, so here's the DNA, one strand, and here's the other DNA. And we already understood, hopefully, that one strand will be produced continuously, right? Okay, continuously in this direction. And then the other one is going to, well, still, it's also going to be produced continuously this way. Sorry. Okay, but then you're gonna have a bunch of Okazaki fragments here, right? Okay, same thing over here. Right. So that brings out an interesting point. The leading strand actually becomes the lagging strand on the other side, and vice versa. Okay. Let me just put some numbers here to help you make sense of it. This is 3 prime. Nope. This is 5 prime to 3 prime. And then this would be, again, like 5 to 3. Okay. 3 to 5. And then 5 to 3. Okay. Now, these purple things, those are the primers, right? Okay. Again, you don't have to copy this. There is a nicer picture in the notes, I'm sure. All right, so here we go. Let's revisit what we just talked about a moment ago. The DNA polymerase 3 will keep on going and going and going until we hit the purple part here, right? Okay, then the DNA polymerase 3 will fall off, and DNA polymerase 1 will come, and it will replace this purple DNA with purple RNA rather with actual DNA. Okay, this RNA will be replaced by DNA because another polymerase will come here, and so on and so forth. But we're gonna have a problem because eventually we're gonna get to the end of the DNA, right? What is going to replace this chunk of RNA, right? We don't have a polymerase 3 coming from upstream anymore, right? So since no DNA polymerase 3 is coming, so to speak, this part will not be copied. Do you guys understand that? Right? This part is going to be replaced with DNA because a polymerase 3 is coming, right? There's polymerase 3 coming to replace this. Polymerase 3 coming, right? When I say the polymerase 3 is coming, what I mean is the polymerase 3 will come, it will fall off, and that would prime the polymerase 1 to come and replace it. But we cannot do that at the end since there is no polymerase 3 coming from upstream, right? Same thing over here, right? Okay, like this part is not going to be copied, right, for the exact same reason. Does that make sense? So each time the DNA replicates, we lose a bit at each end. Okay, it's kind of scary if you think about it. Right? It's like chopping your DNA shorter and shorter and shorter. Okay, so if you think about your chromosome here, right? If you replicate, it's going to get shorter and shorter and shorter and shorter. Okay. So luckily, to counteract this shortening effect, located at the end of the DNA, we are going to have these filler DNA. Okay, we call them telomeres. Okay, they are just repeats of TTA GGG. Okay, it's a bunch of these repeats. Okay, so every time the cell divides, the chromosome will cut a little bit of the telomeres and cut a little bit of the telomeres. Right, so we don't cut into the critical information. Right, so the telomeres get shorter and shorter and shorter. And when the telomeres get critically short, then the cell will not pass G1 and it will not be allowed to go into mitosis. Right? The cell then become growth arrest. It's going to enter a state of senescence. Tyler asks, because it's at the 3' end and DNA polymerase can only, exactly, right? It can only go from the 5 to the 3. So over here, right, we're not going to be able to go backwards to copy this chunk, okay? And we don't have another polymerase 3 coming off, okay? Now, you might be thinking, well, how about this? How about this? How about we put a primer like right at the edge, okay? And then we can copy. Yeah, that works, but still, you will still lose some because this little bit, that primer, even if you put it at the edge, that part will not be replicated because there's no polymerase coming from upstream. Okay, so for that reason, the chromosome gets shorter and shorter every time it divides. Okay, and again, when the telomeres get critically short, the cell will not be allowed to divide again. Okay, so let's say we are born with this much telomeres at the end of our chromosomes. Okay, and you know, every time your cell divides, it gets shorter, 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 shorter. So that means when you're an adult, right, your telomeres are much shorter compared to when they were when you were a baby. Okay, now let's say you want to have a kid, you want to have a child or children. Okay, when you make the sperm or the egg, you need to reset the telomeres. Okay, because if you do not reset the telomeres, your offspring, instead of being born with long telomeres again, if they inherit the shortened telomeres that you have, then they won't have as many divisions in their cells compared to you, right? And then if that keeps on going for several generations, then you know you cannot really live anymore because the moment you're born, then your cell is already at a cellular senescent state, right? Which is why in the germ cells, in the cells that's going to become the sperm and the egg, there is an enzyme that is called telomerase. Okay, so telomerase, which is only, normally speaking, should be activated only in germ cells as well as stem cells. Their function is to, let me find my color here, is to add TTAGGG to telomeres, right? To re-lengthen. Them. Okay, so that when you pass on your genetic material to the next generation, they would have fresh telomeres again to start with. Okay, and then they would erode again throughout their life. Does that make sense? Okay. So one of the limiting factors of why cells cannot divide indefinitely is because of the shortening of the telomeres, right? Okay. Once it gets to critically short, you're not allowed to divide anymore. Now, you might be thinking, hey, why don't we activate telomerase in all the cells? Then they could keep on re-lengthening the telomeres, and then the cells can divide more and more, and maybe we can live longer. Okay. That actually is not a good idea because in cancer cells, it turns out, which is what I did in my grad school study, I look at the expression of telomerase in cancer cells, and that is actually elevated, right? By activating telomerase in normal cells, it causes the cells to become virtually immortal. Okay, these cancer cells can just divide and divide and divide. Did you know if you take some cancer cells and you put it in a petri dish, you can virtually divide them, you know, forever. If you keep on feeding them with nutrients, they would just be dividing and dividing and dividing. They do not stop because their telomeres do not get critically short, right? Because of the overexpression of telomerase. Okay, before we take the break, any question at all? That is all for DNA replication. And when we come back, we're gonna do mitosis, I believe. But once again, any questions? You guys are very quiet. It's hard for me to gauge without seeing your face whether you understand or not. It's one of the drawbacks of online learning. I don't get to read people's faces. Okay, great. If you thought of anything, please leave me a message. Otherwise, I'll see you in 5 minutes. Okay. All right, let's keep going. Question number 8. Identify and briefly explain the function of each cell structure shown in the diagram on the right. Okay. Let's keep going at this thing. Oops, I chose the wrong thickness. A, where is A? A is over here and it is pointing at the chromosomes. Okay. Chromosomes, okay, which of course contains genetic information. Okay, genetic code, if you would. We'll learn how to read the code next week. Okay, it's quite exciting. B is pointing at this black dot location. Okay, that is the centromere. Centromere. Okay, which is the location where duplicated DNA stick together. Okay. C is the centrioles. We learned that. When did we learn this? Lecture 3, centrioles. Some textbook might say like centriosomes. We're not going to make that distinction. It doesn't matter. Okay, so centrioles, this is where spindle fibers are produced. Okay, and the function of the spindle fibers is to help guide the movements of the chromosome. D, I think that's the cell membrane. Okay, function, you already know we had a whole lecture on cell membrane. Okay, I'm not gonna write anything. And the E here, E is the spindle fibers. Spindle fibers, which is, like I said, help guide chromosome movements. All right. Let's look at some poll questions. We have not done any today yet. Let's see if you know the answer to this one. Go ahead. Okay, that was a minute. Just gonna wait a few more seconds for people to lock in their answers. Okay. Yeah, most of you got it. Okay. The answer is B. But let me explain quickly. Prokaryotes have telomeres and eukaryotes do not. Prokaryotes meaning like bacteria and like simple well, bacteria, okay, mostly. Okay, prokaryotes are bacteria and they do not need telomeres. And the reason for that is why. And can anybody tell me why prokaryotes do not need telomeres and eukaryotes like us, we do? We know that telomeres protects the ends of a DNA, right? As they erode. But how come prokaryotes do not need that? Okay, 11 of you did not choose this answer. So does anybody want to explain to me why prokaryotes don't need telomeres? Tyler said circular DNA. That is absolutely correct. Okay, so prokaryotes have circular DNA. So what that means is if your DNA goes in a circle, right? Let's just say you have a primer here, right? Okay, and then you can just keep on going, replicating, and then you just loop back on yourself, and then you can replace it, right? We, as eukaryotes, we cannot do that because we have linear DNA, okay, and the end of it never gets replicated, and so we need the telomeres. Okay, B, prokaryotes chromosomes have a single origin of replication, whereas eukaryotes have many. That is actually true. Okay, circular DNAs are actually quite small in prokaryotes, so you just need to open one bubble and you be done, fairly quickly. Cells can divide, bacteria can divide in as little as 20 minutes in some optimal conditions. Okay, but for us, the chromosomes are so long. If you just have one single replication bubble, it will take forever to copy our DNA. So you need to do it at multiple sites. Prokaryotes chromosomes has histone, whereas eukaryotic chromosomes do not. It's the other way around, and I'll explain what histone is in a second if you don't know. And this is also the other way around. Okay, there is no Okazaki fragments because of the circular DNA condition that we talked about. Okay. So now is a good time to kind of maybe help you understand the difference between the various forms of DNA. Okay, so DNA, you know by now, you have to know by now, DNA is just referring to the double helix, right? Double helix, okay, with the A, C, G, T, okay? And that is really, really long, right? There's no way in its innate form, if you would, that it would fit in the nucleus. So what happens is, in order for us to fit the DNA into the nucleus, we are gonna have some DNA plus what we call packaging protein. Packaging protein. And one example of packaging proteins is what we call histones. Okay. Histones are positively charged, and that is perfect for binding DNA because if you think about the backbone of the DNA, it's made up of the sugar and the phosphate, right? Phosphate is PO4 3-. So the positively charged histone is going to be attracted to the negatively charged backbone. Okay, and then the histone and the DNA together is going to cause the DNA to condense into like a bundle. Okay, and so this more compact version of DNA with the protein, we call that chromatin. Okay, chromatin. And then it can condense even more, contains more. Okay, so this stuff is going to coil up even more. Okay, like this. And this is the super condensed version. That's essentially what these things are. And that is your chromosome. Okay. This super condensed form makes it easier to move the DNA around without tangling them. Okay. So this only happens after DNA replication and during the mitosis. Right? So we have DNA in its, purest form. We can package them to form chromatin, and then if they condense even more, then they get chromosomes. Okay, and each half of these, that's the chromatid. Okay, some people pronounce this like chromatin to distinguish it from chromatid. Okay, I don't know. Say whatever you want. I'm not very good in all these English stuff. So that is that. All right, let's move on to our mitosis right here. Okay, now end of interphase. This is not part of mitosis, right? Not part of mitosis. Okay, this is like after S phase, right? And maybe G2. Okay, so this is a cell that is ready to divide soon. Okay, so what's going on here? Well, by this point, right, DNA replication is completed. Okay, so all the stuff that we learned in the first half, right, that the helicase, the DNA polymerase 3, those things would have done its job, right, and DNA replication is completed. Okay, and what happens is the thing is still, like a mess. The DNA, you can't really see the individual chromosome. So what happens is the chromatin starts to condense into chromosomes. Okay, and then basically it is ready to go into prophase. Okay, now this is the first step in mitosis. Okay, P is for preparation, if you would. It's preparing to move the chromosome and so on and so forth. So what's happening here? Well, a couple of things. Chromatin are fully condensed into chromosomes. Okay. Replicated DNA are joined at the centromere. Okay. Centrioles produce spindle fibers. And start to migrate or move to the opposite pole. And finally, nuclear envelope starts to dissolve. Okay, so the chromosomes can come out, right? When all that is done, we're going to move on to metaphase. Okay. So in the metaphase, spindle fibers attach to the centromere of each chromosome. Okay. a tug of war between opposing spindle fibers will line up all the chromosomes in the middle of the cell. And we have anaphase next. Right? A is for apart. Right? So spindle fibers separates. Sister chromatids and move them to opposite poles. Okay, so these sister chromatids, right, of each chromosome pair, they get pulled apart and they will move to the opposite side. And then telophase is basically the reverse of what happened at the beginning. Spindle fibers disappear. Nuclear envelope reform. And then chromosomes uncoils, right? Back into the chromatin. And also we have cytokinesis, right? Which is the division of the cytoplasm, by creating two separate cells. And it's basically the telophase. All right, it's pretty straightforward. Nothing too complicated, I hope. Okay, let's do more polls. Go ahead. And that has been a minute. Anyone else? Okay. 3 more people. Are you guys there? Okay. So a cell has 8 chromosomes in metaphase. How many chromosomes will there be during anaphase? Let's go back to this picture here. That is metaphase right here, right? Okay, that's metaphase. How many chromosomes are here in metaphase? Okay, so remember, when they're stuck together, we still count them as 1. So that's 1 chromosome, 2, 3, 4. 4 chromosomes here. Okay. And then we're gonna take a look at here. How many chromosomes do we have at anaphase? They're no longer stuck together. So that is actually 1, 2, 3, 4, 5, 6, 7, 8. Okay. So if you have 8 chromosomes in metaphase, how many are you gonna have in anaphase? 16, right? Okay. Hope that makes sense. For those of you who chose B and A, have I convinced you that it is 16? Will you be choosing 16 on the test? Or do I need to explain more? Okay. How about this one? This should be straightforward. Okay. The correct answer is telophase, right? That is where you will do the actual division, the cytokinesis, creating 2 daughter nuclei. Let's try this one as well. Yep. All of you chose chromosome. That is correct. Okay. This is application question. Okay. Not sure how to pronounce that, but this drug inhibits the formation of spindle fibers. Okay. Taxol maybe. Explain why it is used as chemotherapy drug for cancer treatment. What might be the side effects? Anybody want to take a shot at that before I explain it? How does inhibiting the formation of spindle fibers be used as a chemotherapy drug? Okay. So cancer, if if you summarize it in a simple sentence, is basically uncontrolled cell division. Okay, and Adriana said the drug, right, the paclitaxel, it is going to stop the cell from dividing, right? By inhibiting the formation of spindle fibers, there is nothing to move the chromosomes around, and the cells cannot divide, and that will help stop the cancer from progressing, right? It will stop it from dividing and the tumor getting bigger. But of course, normal cells also divide. So the side effect is going to be there will be a suppression of actively dividing cells in the body. Okay, so things that divides rapidly in the body, including like hair follicle, for example. So you might have heard of people undergoing chemotherapy. They might lose hair, right? They might become anemic because you will learn this next semester, but we have stem cells in the bone marrow that produce new red cells, new white cells, and those cells are actively dividing, the stem cells in the bone marrow. So the chemotherapy drug might cause them to not be able to do that, right? So if you don't have enough red cells, you become anemic. Okay, your immune system might weaken, right? As a result of that as well, because you're not producing enough white cells. Okay, but the bottom line is by inhibiting formation of spindle fibers, you are going to inhibit cell division. Okay. We have a few more questions here, but for the interest of time, I'm going to go back to this for a second. I just want to know, does anyone have any question about these ones? Any requests for 1, 2, and 3 before I take up the application question? Any requests? To be. What is the difference between the N15 medium and the N14 medium? Okay. So nitrogen is a key component in DNA, right? Because the nitrogenous base, right? Okay. So N15 and N14, they are basically two isotopes of nitrogen. Okay. And isotopes are basically atoms of the same element, but with different mass, okay, because of the difference in number of neutrons. So nitrogen 15 is a little bit heavier than nitrogen 14, right? The purpose of this is to be able to make the DNA weight different. So if the DNA has a lot of nitrogen 15 in it, it's going to be heavier. So when you spin it down, it's going to sink to the bottom. Whereas if you have a lot of nitrogen 14 in it, it's going to flow to the top. And if you have a mix of 15 and 14, it's going to be somewhere in the middle. Okay. So what they did is they grow the bacteria in the heavier medium for several generations first before transferring it to the lighter medium. And the purpose of that is to ensure that all the E. coli in the dish is going to start with N15 because they have been growing in the medium with N15 for several generations now. So in theory, a majority of the DNA is going to contain the heavier version of the nitrogen. Okay. And then you transfer them to the lighter one. So from that point on, all the nitrogen that will be used to build new DNA will have the lighter version instead. Right? And that's how you can detect the difference. Does that answer your question? All right. Anyone else have any other requests? All right. If you think of anything, a 2D, yeah, Bianca said 2D. Draw the results expected if DNA replication is conservative, semi-conservative, or dispersive. All right. Let me, instead of drawing it, because every time I draw it, it looks very messy. I'm just going to copy it from the PowerPoint, and then I'm going to explain it. Okay. So it's less messy. So here, the pink The pink is the original DNA. These are the N15, the heavy stuff. Okay. So if I take this and then I spin it down in the test tube, right? You will see a single band at the bottom. Okay, because it's heavy, it's going to sink to the bottom. Okay, and then we can transfer this to N14 and grow them, right? Yeah. So if it's the semi-conservative model, what that means is you are going to separate the two parent strand, right? So one pink parent strand on each of the new chromosome, and then you're going to build another one with N14. Okay. So if I am going to take this sample and I spin it down, right? Instead of having a single N15 band in the middle, we're going to have a band that is at the bottom, rather. You're going to have a band that's in the middle. Okay, so that's like a N15, N15. I'm just going to put 15, 14 band. Okay, somewhere in the middle. Okay, so that's the semi-conservative. Half is old and half is new. Okay, and we're talking about the entire one entire strand of DNA is old and the other half is new. Whereas in the conservative model, right, if this were true, then instead of having half and half, you would have the entire original DNA, right, untouched, and then you're gonna make a completely new copy. Okay, so you can think of this as photocopying. Okay, you have your original copy, put in a photocopy, and now you have two copies: one original, one new copy. Okay, if you think about the semi-conservative model, it's a little bit weird. It's like this. Okay, you take the original copy, you photocopy it, right? So now you have two copies, right? But instead of having one old, one new, you tear it down right in the middle, and you stick the original and the copy together. So that's one copy, and then original and copy together, and then you get another copy. Okay. So if I were to spin this down, what do we get here? If I spin this down, I would have a pink band at the bottom, and then I would have a blue band at the top. Okay. And then dispersive is, rather than having exactly one strand new and one strand old, the old and the new are kind of like dispersed throughout the DNA. Okay, so it's not half, it's still half and half, but not restricted to one side of the DNA strand. Okay, it's kind of all over the place. So if I were to spin this down, I would also get a 15-14 band in the middle. And that's what they detected. Okay, after one generation of division, they spin down the DNA, they saw the, this is the result. Okay, which is why they cannot decide whether it was a semi-conservative model or the dispersive model. They knew it was not this one because they did not observe two DNA bands. They only observed one middle band, okay, which is why they need to do it one more time, okay? And when they do it a second time, right, and you can see following the semi-conservative model, you would take away the blue part, and then you would grow another entirely new blue strand, and then the pink one would get another blue strand. So over here, you're gonna get a middle band, right? You're gonna get a middle band that represent this DNA, and then you're gonna get a blue band at the top that represent the lighter one. Okay. Whereas over here, if dispersive was true, then you know this after the second division, it's still kind of all mixed here and there, and you will still observe a single band in the middle. Okay, they did not observe a single band in the middle after the second division, and that's how they concluded it was the semi-conservative model. All right. Hope that answers your question, Bianca. Any follow-up questions? Okay, thank you. Application question. Answer the following question based on the karyotype shown on the right. This is a karyotype, right? A karyotype is an orderly arrangement of chromosomes, right? Extracted from your DNA, from your cells, rather. So which chromosome is the longest? Which one is the shortest? So these are arranged based on the length, and you can see chromosome 1 is the longest. Okay, so chromosome 1 is the longest, and the shortest is chromosome 22. Okay, Ashana, can you explain what? Yeah, I'll go back to 1E after. Okay, let's finish this first. How are chromosomes numbered? Okay, they are numbered based on size and position of centromere. Okay, so you can see this is the longest, right? And then the position of the centromere also shift as you go down the chromosome number. Okay, so obviously I'm not a trained cytologist, so I wouldn't be able to explain to you how exactly they line this up, but I know it's based on these two things. Is this male or female? How do you know? Anybody? Any takers? Male or female? Yep, this is a guy because of the XY, right? Genetically, this person is male. XY is male. Female is XX, right? Okay, so this is a male. Explain whether the karyotype shows any abnormalities. Okay, so anything wrong with this? See this picture? Any abnormalities? Yep, Tina says the number of chromosome 21, there's an extra chromosome 21, right? Okay, so we call this trisomy 21. And having 3 copies of chromosome 21 leads to Down syndrome. Okay, so there you have it. Now, who would be interested in doing a karyotype? Actually, parents who are at risk of certain genetics conditions, they can do what's called amniocentesis, right? Did I talk about this in the lecture? I feel like I have. Did I? Anybody watch it? Amniocentesis? No? Okay. So amniocentesis, I think that's how you spell it. Any mothers here who might have been informed about amniocentesis? Want to explain what it is? Tina, you mind explaining it, or you rather I explain it? Oh yeah, please go ahead.
Sure, I can. So when I was pregnant with my 4th child, I had, I guess they call it pre-screening testing.
Yeah.
And certain markers came up in my blood test and my ultrasound, which then they said to have an amniocentesis, which is a needle that goes in through your tummy into your uterus and takes a sample of the amniotic fluid and tests the chromosomes of the baby.
That is exactly right. Thank you so much for explaining and sharing your story. So for those of you who are not familiar with this, the baby, the fetus, grows in the uterus of the mother, and there is an amniotic sac that surrounds the baby. It's kind of like the egg yolk of an egg, if you would, okay? And inside that sac is a lot of fluid, right? That keeps the baby floating. That fluid is called the amniotic fluid, okay? That fluid contains a lot of the baby's cell. And so what happens is I think at around week 21 or so, they could, if the mother wishes to have this done, insert the needle through the belly button area, and they would draw a bit of this fluid and extract the cells from the fluid, the baby cell, and that way they could look at the baby's chromosomes, right? And one of the things that they could detect is, for example, Down syndrome. Right? If there's extra chromosome 21, then they would be able to inform the parents, and then how to proceed. Of course, that is a big ethical debate, but it is something that can be done. Of course, as Tina mentioned, not everybody's going to do amniocentesis, right? This is usually something that will be done only after certain markers have come up in the prenatal screening. Okay, so there are blood markers that they could check. There are ultrasound markers that they could check, but they could do a little ultrasound to look at, for example, the baby's brain. Right? If certain areas are more what they call like echogenic on the ultrasound, then that could be a markers. And if you have a lot of these markers, right, compound with age and family history, if you are at risk of certain genetic disorder, that could be detected by amniocentesis, that would be something that that could be done. Okay, so I hope that explains it. And what was it before? Hashana said 1E, right? Is this one, right? Why were radioactive sulfur and phosphate used in the experiment as opposed to other elements? That's because sulfur is uniquely found in proteins and not DNA, right? Remember, proteins are made up of like CHNO and trace of sulfur, right? So that sulfur is found only in the proteins and not the DNA. Okay, so by having a radioactive sulfur, if I detect the radioactive sulfur, I know I'm detecting protein. Okay, phosphate, on the other hand, is found in the DNA and not protein, and that's why these elements are chosen. Okay, any other question? Okay, let's see if there are other practice questions worth doing. I think these are pretty straightforward. Okay, maybe we will. You know what? Maybe I'll just copy them, and then I will post it along with this thing, and then you guys can try it out. Okay. In the last couple minutes, let's fill in some of these things. Okay, fill in the blanks. You know what? I'll post a fill in the blanks as well, and we will take up this diagram over here. Okay. So, you know what? Let's take up this one as well, because it looks very messy. Help me. Feel free to help me out. I'm always happy when you guys participate. This one right here. What do you think this enzyme is? This one right here. You know what? Maybe let's start with this one. This one is probably easier. This right here. What do you think this one is? Number 2. Excuse me. What is number 2? Anybody? Yes, Tyler is correct. This is the helicase. Okay. Other people also answered. Very good. This is the helicase, which helps unzip the DNA, creates a replication bubble, and it's going to be going into the fork. As the helicase unwinds the DNA, you're going to build up tension in the DNA strand, and we do not want too much tension. So we're going to need this enzyme, and that is, of course, what is that? Gyrase. That's right. The DNA would have a tendency to reanneal, have a tendency to come back together. And we don't want them to come back together. We want to keep them apart so that we can make a complementary strand. So to do that, we need the single strand binding proteins. Okay. Now, this is a black and white photo and a diagram, unfortunately. But number 5, or sorry, rather, let me see right here. This dark, a little bit darker here, right? Thing here, that's going to be colored in pink, right? So that it helps us to do it. Does this one have one? Probably here. I'm just going to make that up. So all these pink things, right? All these pink things, they are the RNA primer. The RNA primer is created by the RNA primase. RNA primase. Okay. And the purpose of the primer is to help initiate the DNA replication process. Okay. So over here, right, we have our DNA polymerase 3. Number five is just pointing at the leading strand, because that's the strand that is being produced continuously. Leading strand. And over here, we can see these are being produced in fragments. Okay, so they don't have a label for that, but I'll add it in. Those are the Okazaki fragments. Okay. Oh, I guess they do. I guess that's what 5 and 7s are. Okay, the Okazaki fragments. And over here, that would be the ligase. The ligase that helps connect the strands together. Okay, what color have I not used yet? There we go. Ligase. Right here. Okay. Again, pretty straightforward. Let's try to identify the phases here. First one here. This is a plant cell because of the rectangular shape. And I cannot really see the chromosomes yet. They haven't really condensed yet. And there's only one nucleus. So I'm going to say this is interphase. This one, you can see it's already starting to split. And so it is telophase. Over here, I see the spindle fibers. I see that the nuclear envelope is starting to dissolve and the chromosomes are fully formed. So I'm going to say this is prophase. Chromatids are separated. They are being pulled to opposite ends. And so this is anaphase. Over here, same thing, nuclear envelopes is disappearing. The centrioles are moving to opposite side, and we can see the fully formed chromosome. This is the prophase. Note that there are no centrioles, right, in the plant cells. Nevertheless, the spindle fibers will still form. This one should be apparent. It is anaphase. Everything is lined up in the middle. Metaphase. Metaphase again. Two separate nuclei, and it's starting to divide. So this would be telophase. And the first real picture, you can see the chromosomes are being pulled apart here. So that is another anaphase. Is it okay? Does anybody have any question? Let me see if there's any important things coming up before I let you go. I'm just gonna see the calendar for a second. You guys are 2:02, right? Yep. All right. It has already been halfway in the course, if you can believe it. We just did the test last time. So, okay, nothing yet. But the lab is going to be due on the 28th. Okay? The lab is already available. So if you want to get a head start on that, you could. There's going to be a simulation on how to use the microscope and stuff like that. But I will give you more details later on. Looks like we still have time. So I probably will explain the lab to you more later. There is the last day to drop on the 18th. So if you drop the course before that day, then your marks are not going to be showing up on the transcript. Okay, so after that day, you know, if you fail the course and at the end, it will show up on your transcript. Or if you decide to drop before the finishing the course after that day, then it will show up on your transcript as like, you know, withdraw from the course. Okay, so some people don't want to have that on the transcript, and if they feel like they're not doing too well at this point in the course, they might want to drop it. But dropping the course will take you out of any articulation that the program offers, right? So, you know, before you drop the course, maybe just send me an email, and discuss options, right? Or talk to Martin to see what your options are. Sometimes you don't have to drop the course. Sometimes you think you're doing bad, but in fact, you are not really doing bad, okay? But of course, all the marks are posted on eCentennial, right? There is also like a grade calculator that you could use to see where your standings are and what you need to get in the course in order to get a certain final grade. And I'll post a link in today's debrief for you. Okay, so next week, no evaluations yet. Okay, just lecture 7. That is a particularly difficult lecture if you haven't learned it before. So I encourage you to watch the recordings before coming to the tutorial. Okay, so thanks again for coming. I hope you guys have a good week, and I'll see you all next week. Bye for now.